Welcome back to this next video, and this is the part 2 on the transport of material across the cell membrane. In the last video I told you about the active transport, and in this particular video I'll be focusing on the passive transport with examples so that you can get a better understanding of the passive transport. In the last video I told you that there are actually three types of the porters. One is known as the uniporter, which is actually moving one substance in one direction, whether that is moving inside the cell or that is moving outside the cell. In the synporter, two of the substances, they are moving, but they are moving in the same direction, whether both of them are moving inside the cell or both of them they are moving outside the cell. And in the antiporter, I've told you that one of the substances is moving inside the cell and the other one is moving outside the cell. Then I've told you that there are two major ways that a molecule can move across a cell membrane, and the distinction has to do with whether or not cell energy is used. In the last video I've told you that when you talk about the active transport, you need the energy in the form of the hydrolysis of the ATP, and the hydrolysis of the ATP actually moves the material in a particular direction. But in the passive transport, no energy is required, and the substances, they can actually move passively without utilizing the energy. So when you talk about the passive transport, it is actually a type of the membrane transport that does not require energy to move substances across the cell membrane. Now, instead of using energy like that we see in the active transport, in the passive transport, the substances are moving from an area of high concentration to an area of low concentration, and this movement is favored because this movement actually increases the entropy of the overall system. Now, the rate of the passive transport, that depends on the permeability of the cell membrane, which in turn depends on the organization and characteristics of the membrane lipids and proteins. When you talk about the types of the passive transport, there are broadly four types of the passive transport. One is known as the simple diffusion, the other one is known as the osmosis, the third one is the facilitated diffusion, and the fourth one, that is the filtration. So let us discuss them one by one. When we talk about the diffusion and osmosis, usually these two terms they are actually discussed simultaneously. When you talk about the diffusion, it is actually the movement of the molecules from high concentration to low concentration. Same is true for the osmosis, that you will be moving substances from higher concentration to low concentration. As you can see over here, this is one of the solution where you are actually having a high concentration of the solute, and in this particular case the solute, they are actually moving towards the area where there are low concentration of the solute, and this movement continues until the solution get equalized. Same is true with the osmosis, in which the movement of the solvent, especially the water molecules, they move across a cell membrane from high concentration to low concentration. If you talk about the differences between the two, as you can see over here, in diffusion you actually do not need any semi-permeable membrane, but in case of osmosis we do need a semi-permeable membrane. So this is one of the basic differences between the two, that in case of the osmosis the movement is from the higher concentration to low concentration but across a semi-permeable membrane, but in case of the diffusion, the movement from high concentration to low concentration, you do not need a semi-permeable membrane. That is one of the difference between the two. Another difference between the two is that in diffusion, both the solute and the solvent, they can move, but when you talk about the osmosis, we only talk about the movement of the solvent, and in most of the cases this solvent, that is water. So these are the two differences, two major differences between the osmosis and the diffusion, that you do not need a semi-permeable membrane in diffusion, and both the solute and solvent can move, and in osmosis you need a semi-permeable membrane and only the solvent moves, usually the solute do not move. One of the important example of the simple diffusion in the human beings, that is the pulmonary gas exchange. When you talk about the respiration system, and when you inhale air, there is a high concentration of the oxygen in the alveoli. So the blood that is moving towards the alveoli, that is having a high concentration of the carbon dioxide. So as the alveoli have a low concentration of the carbon dioxide, the blood has a high concentration of the carbon dioxide. So the carbon dioxide is going to move into the alveoli. Opposite to this, in the alveoli there is a high concentration of the oxygen, and in the blood there is a low concentration of the oxygen. So the oxygen is going to move from the alveoli into the blood, that will then be distributed to different parts of the body. So this is simple diffusion, where the substances, where the gas that is moving from high concentration to low concentration. When you talk about the osmosis, we usually see that there are three types of the osmotic solution. As I've told you, that in the osmosis we are actually focusing on the movement of the solvent, especially the water. So when you talk about the types of the osmosis solution, there can be three types. One is known as the hypertonic, the other one is known as the isotonic, and the third one is known as the hypotonic. So let us discuss them in detail, what these terms mean. Now in the isotonic solution, the concentration of the solute that is equal inside and outside the cell. And in the isotonic solution, as you can see, the water is going to move inside the cell, the water is going to move outside the cell, but they are in equal quantity. Say for example, 2 molecules of the water, they are moving in the cell, so 2 molecules of the water, they will be moving outside the cell. So this iso actually refers to same. So as the concentration of the solute inside and outside the cell is the same, therefore the movement of the water, that is equal. This is known as the isotonic solution. When you talk about the hypotonic solution, in the hypotonic solution, the concentration of the solute is high inside the cell and that is low outside the cell. So when the concentration of the solute is high inside the cell and that is low outside the cell, the water is going to move inside the cell because there is a high concentration of the solute. And in these particular cases, the cells, they can actually burst. So the cell, they are going to burst in the hypotonic solution because a lot of water is going to move inside the cell. When you talk about the hypertonic solution, in the hypertonic solution, the concentration of the solute that is high outside the cell, that is low inside the cell. So the water is going to move from the cell into the exterior environment, and there will be a shrinkage of the cell. So this is how the red blood cells, you can expect them in the isotonic, in the hypotonic, and in the hypertonic solution. And as the water is moving through the cell membrane, therefore these are examples of the osmosis. The third type of the passive transport, they are known as the facilitated diffusion. And in the facilitated diffusion, as you can see in this particular image, there are ion channels, or there are protein channels, and there are carrier proteins. Again, the substances, they will move from higher concentration to low concentration, but they will be needing membrane proteins, like as you can see over here, or they will be needing some carrier proteins. When you talk about these protein channels, they are actually the transmembrane protein, and what I mean by that is that these protein channel, they are moving from extended from one end of the lipid bilayer to the other end of the lipid bilayer. So they are actually across the cell membrane. So the solute, the substances, they will move with the help of these protein channels from the area of high concentration to the area of low concentration. As you can see over here, these green substances over here, their concentration is high in the extracellular space, their concentration is low in the intracellular space. So they will move from the higher concentration to a low concentration, but this, in the facilitated diffusion, the movement will be through the protein channel. Sometime you will be talking about these carrier proteins, and what these carrier proteins do, that they are actually increasing the affinity of the substances for these protein channels, and when they increase their affinity for these protein channels, they actually move from higher concentration to low concentration. As you can see, these structures over here, their concentration is high outside the cell, that is low inside the cell. So these carrier protein are actually increasing their affinity to be moved moved from the exterior into the interior of the cell. The last type of the passive diffusion or the passive transport, that is known as the filtration. And filtration is actually the movement of water and solute molecules across the cell membrane due to the hydrostatic pressure generated by the cardiovascular system. Now, depending on the size of the membrane pores, only solute of a certain size may pass through it. You may be having a variety of the solutes over there, but only those solutes of which have got a specific size which can pass through the membrane, they will be allowed to move to the other end. For example, when you talk about the membrane pores of the Bowman's capsule in the kidneys, they are very small. These pores are very small, and they only allow the smallest of the proteins like the albumin. Only the albumins, which are the smallest of the protein, they can actually move through these small pores, and they have any chance of being filtered through. This is, say for example, a filter. This is the process of the filtration in which you give it a feed. As you can see over here, the smaller size, they can actually cross this membrane, but the bigger particles, they are actually stuck in this filtration process. So when you give them a feed, only the substances of a particular size, they can actually move, and when they move to the other end, they are actually known as the filtrate. So these are the 4 types of the passive transport. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.